Hello dear students, welcome to the another video tutorial of mammalian histology. Okay, so in last session we have seen the alimentary canal and the glands associated with the digestive system. Okay, so in last session we have seen the three type of the salivary glands. Okay, the salivary gland will be the parotid, then submandibular and sublingual. Okay, so in this session we will see about the two other glands or the two other organs which are going to give their um, somewhat uh, their specific for the supporting glands for the digestive system. Okay, in that the first gland will be the liver. Okay, and then we will see for the pancreas. Okay, so the liver is the largest exocrine gland in the body. Okay, which is basically a compound tubular serous gland. Okay, that means they are secreting some fluids, the serous. Okay, and this is situated below the diaphragm and above the intestine. Okay, as you can see in the picture. Okay, that giving you an idea about a location of liver in the abdominal cavity. Okay, where it is situated below the diaphragm and above the intestine. Okay, and in position by the connective tissue membrane to the at its right hepatic lobe. Okay, okay. So the right hepatic cavity is covered by the liver, whereas your left cavity, just below your the left side ribs, is covered with the stomach. Okay, the pylorus cavities. Okay, which is covered by the pyloric, fundic and the these three regions of the stomach. Okay, then this liver performs n number of functions. Okay, such as secretion of bile. Okay, the secretion of bile salts to maintain our sugar level in the blood. Okay, the synthesis of prothrombin, serum albumin and serum lipoprotein. Okay, then the third function will be the storage of glycogen, fat, protein and vitamins. Okay, then will be the serous diminulis are numerous and the large with the mucosalveoli. Okay, then will be the plasma protein synthesis is also done by these some cells, specialized cells. Okay, whereas synthesis of albumin. Okay, then lipoproteins, then glycoproteins. Okay, then prothrombin and fibronectin. Okay, and that fibrinogen also synthesis in this liver. Okay, then vitamin storage and modifications on that stored vitamins are carried out by means of this liver, like vitamin A, vitamin D and vitamin K. Okay, some sort of iron storage and metabolism also done by the liver, like transferrin, hepatoglobin, hexoplexin. Okay, then the ferritin. These are the some hemopexin. Okay, then the drugs and the toxin degradation, that means detoxification, also done by means of the liver. And the main important uh, part will be the bile production. Okay, then one of the main important part will be it take part in the carbohydrate metabolism. Okay, whereas the liver will be the large essential organ found in the upper right quadrant of the abdomen. Okay, so as you can see, this through the liver performs diverse functions. Okay, they performs n number of functions. Okay, and then it has no group of cell cytologically specialized to perform one particular function. Okay, and these other as pancreas. Okay, pancreas having the two type of the cells. Okay, whereas it having the uh, exocrine secretions also as well as the endocrine secretions also. But here in case of the liver, it cytologically not having any not having the preferred uh, cells to produce to synthesis a particular type of the enzymes or the secretions. Okay, then it also exhibits, the liver also exhibits number of structural features which going to differ from the other gland. Okay, so the liver color is reddish brown in color. Okay, and several fissures partially divided into the four lobes. Okay, we will see that lobes in next coming slide. Okay, the microscopic structure of liver. Okay, as you can see, the liver is incompletely covered by the outer tunica serosa derived from the peritoneum. Okay, and below it, liver is completely covered by the delicate connective tissue capsule are named as the capsule of Gilson's. Okay, and these capsules contain abundant elastic fibers. Okay, which capsule? The capsule of Gilson's. Okay, and at one place on the lower surface of liver, that connective tissue going to enter in the substance of the liver to form the septas. Okay, the some gap in between two specialized cells. Okay, and that partitions or that septa which separate group of liver cells from the liver lobules. Okay, with the place where the connective tissue going to enter in these septas called as the aorta or the hilum. Okay, and the septa going to carry the blood vessels, nerves and the lymph vessels. Okay, and the connective tissue forms the framework in this gland. Okay, so the hepatic lobules, that means the lobes of the liver. Okay, the hepatic lobules or the hepatic cells are arranged into minute irregular portions that having the polyhedral blocks called as the liver lobules. Okay, or the hepatic lobules. Okay, so which are going to transverse sections look like somewhat the hexagonal in shape. We will see the transverse section or the TS. In that, these hepatic lobules or the hepatic cords are looking like the hexagonal shape, not perfectly hexagon, but it having the uh, sections. Okay, as you can see in the picture, it having the right lobule and the left vebule, which is separated by means of this septa or the tendons of the levers. Okay, and these right and left quadrate or the lobules. Okay, so it is the multifunctional accessory to the gastrointestinal tract. Okay, the liver is multifunctional accessory. Okay, so it performs duties like detoxification, protein synthesis, biochemical production of nutrient and storage of them, as well as process on the storage nutrient or the vitamins. Okay, so the there is no cytologically specialized group of cells are present for the particular function in the liver. Okay, as I said, its color is reddish brown and partially divided into four main lobes. Okay, as you can see, the right, left, and the quadrate lobes. Okay, as you can see, which is highlighted in the green color in this picture. Okay, so these hepatic lobules, or these, each lobule in human is about 2 mm tall and 1 mm wide. Okay, so the lobules are more distinct in, uh, you can see in the cross sections, like animal like the pig, rat, cat. Okay, which uh, because they look in them, because the connective tissues, the Gleason's capsule, that means, okay, that going to investment, going to passion that investment and that connective tissue. Because of that, these lobules are more distinct in the pigs, or the cat, or the rat. Okay, so the spaces formed between these neighboring lobules, as you can see, this will be the this is the TS of the liver. Okay, so in that you can see the one section that is highlighted. Okay, as you can see, the it having the hepatic cords, or the parietal zone, as a first zone in between these hepatic lobule. And that hepatic lobule, as you can see, the hexagonal like shape hepatic lobule in this TS, you can see. Okay, in that, the three, it from the central to the periphery, it get divided into the three regions. That will be the peripheral zone, that will be named as the zone 1. Then the intermediate zone, that will be the zone in between the central zone as well as the peripheral zone. And then third zone will be the central zone. Okay, at the uh, center of this hepatic lobe. Okay, at center you will see the central vein is located. Okay, then will be the portal area, the hepatic portal lobe, and the hepatic portal veins are carried out from here to the which is going to open into the duodenum. Okay, and that hepatic portal area, as you can see, then will be the interlobular septum, you can see at just beneath the portal area. Okay, so this will be the TS. Okay, so the hepatic cells and their arrangement in the lobule, as you can see, like that, the liver has two main constituents, an epithelium parenchymal cells at its periphery. Okay, and the system of the blood canals are, you can see, with the portal areas or the interlobular septum. Okay, then will be the cell has a distinct cell membrane. Normally each cell has central nucleus, but two or more nuclei are not rare in this case. Okay, so the cytoplasm contains the numerous mitochondria in them. Okay, and the endoplasmic reticulum, especially the rough appearance is due to the rough endoplasmic reticulum. Okay, they are besides the nucleoproteins or the lysosome, or the glycogen and the fat droplets. Okay, and the bile pigments may occasionally be seen inside this type of the cells. Okay, so the hepatic cells are arranged in the irregular branching and interconnected plates. Okay, which going to appear like cords, where these hepatic cords, as you can see, when the hepatic cords or the hepatic below the hepatic lobules. Okay, and these hepatic plates or these laminae going to form a secretory portion of the gland, okay, whereas the surrounding the hepatic cells will be the forming a network of a fine bile canicules, okay. So the hepatic sinusoids will make up a intralobular system of blood capillaries which going to convey this blood from the portal areas to the central vein, okay, from the uh, periphery to the central zone, okay, towards the central vein, okay, and these hepatic artery going to provide the blood supply, okay. So likewise in zone 1 you will see it is the one which will be the closest, okay, and to having the short axis, okay, as you can see in the zone 1, okay, it having the closest to the short axis, hence the portal canals and the supply of arterial blood is more at the zone 1. Hence there is will be the blood supply will be more at the zone 1 area, at the peripheral outer locations. The hepatocytes in the zone 1 receives the highest amount of the oxygen, okay. At when you going to the zone 2, it is the zone 2 which is located in between the zone 1 and 3, and zone 3 specifications will be it is the one furthermost from the short axis areas, okay, but they will be the closer to the central vein, okay, and this they are hence they are closer to the central vein, okay. These hepatocytes receives the least amount of the oxygen, okay. The hepatocytes are nothing but the cells of this liver, okay. So these hepatocytes are nothing but the large and polyhedral, that means it having the 6 surfaces cells going to make up 80% of the total cells of the liver, okay. Then they can contain between 2 and 4 nuclei, okay. So which are the large and spherical occupying the central surface, and that center of the cell each nucleus has at least 2 nucleoli, okay. That why that hepatocytes having the 2 nucleoli in each nucleus, okay. The typical lifespan of the hepatocytes is having for the 5 months only, okay. So then after 5 months they are get replaced by the their regeneration is continuous in these hepatic cells, okay. Then it is replaced by the new cells, okay. So the cytoplasm is acidophilic over here with the dotted basophilic regions that going to represent by the uh, rough endoplasmic reticulum, it also referred as the RER, and ribosomes. So due to the rough endoplasmic reticulum and the ribosomes, okay, so these will be having the rough appearance in these hepatocytes, okay. So as you can see the blood supply for these bile so for these hepatocytes, okay. So it having the separate bile ducts in the liver, okay, that uh, as you can see in this area, that hepatic lobule, and they are going to having with the at the peripheral zone 1 they are having the liver acinas, they are having the separate bile area or the bile duct in the liver. So the between the cells are minute canals are present that are known as the bile caniculi, okay. So these bile caniculi going to end their frame of network with the and that are these bile caniculi, and that going to open into a small bile ducts. Okay, and the small bile ducts at the periphery of these lobules. Okay, so these latter going to have the larger and larger bile ducts which run along with the pancreas of these uh, latter ones. Okay, and these hepatic arteries and the hepatic portal veins that going to produce a small bile ducts are lined by the cuboidal cells, and they are larger ones that line up by these columnar cells. All larger ducts form the larger ducts. Okay, and going to emerging from the left and right side of the liver. Okay, they come out from the liver and at the hilum. Okay, so they come out from the liver at the hilum to form a common duct. Okay, and blood supply is provided by the liver to which receives from the arterial blood from the hepatic artery and the venous blood from the hepatic portal vein. Okay, so the vein brings blood from the intestine which is going to laden with the carbohydrate, fat and protein. Okay, so the liver cells stores these substances in their cytoplasm. Okay, so the hepatic veins, when they going to drain out the blood supply into the inferior vena cava, at that time the lymph vessels, so these lymph vessels are rich plexus in the Gleason's capsule. So they are providing the septa and also surround the bigger blood vessels. So the nerve supply is present in the liver. Okay, so the nerve supply is given by the non-medullatal type of the nerve fibers that going to derived from the um, uh, derived from the parasympathetic or the sympathetic parts of the autonomic nervous system. Okay, and that autonomic nervous system going to supply to the liver as nerve supplies, and that communication with the um, our uh, nerves. Okay, so they are innervate the blood vessels and the bile ducts from these nerve supply, and they are carried out their uh, interconnections. Okay, so the distinguishing characters or the distinguishing features of the liver, okay, all are compiled in one slide. Okay, so the Gleason's capsule is present. Okay, so the numerous hepatic lobules are seen in the hepatic lobes. Okay, so in the center of the lobule the central vein is present. Then the fourth one will be the hepatic cords are present. Okay, then will be the sinusoids and the portal areas are present in the liver. Okay, and the main function of the liver will be, okay, so detoxification, protein synthesis, biochemical productions and the nutrient storage. Okay, so the liver performs such a functions that drugs and toxin detoxification, bile production, as well as it having the some role in the carbohydrate metabolism. So over than okay, over than 500 so key fact about the liver. So it having the over 500 functions. Okay, in that we have seen the main functions like the xenobiotic biotransformations. Okay, and then will the protein synthesis, the nutrient storage, then bile production and the detoxification. These are the some highlighted functions of the liver, but actual we if you going to study in detail, there are over 500 functions of the liver which is carried out by means of the liver. So that's why it is the largest gland in the human. Okay, the anatomy we have seen it having the four lobes, the right lobe, left lobe, quadrate lobe and the quadrate lobe. Okay, we have seen in the picture. Okay, so it having the two surfaces, the diaphragmatic surface and the visceral surface. Okay, why it is the diaphragmatic, the below the diaphragm it situated at that surface is named as the diaphragmatic surface, and the other uh, visceral organ, that's why it is the visceral surface. The ligaments are present in the liver, that will be the coronary ligament, then the left triangular ligament, facilinular ligament and the round ligament. Okay, so the lymphatic drainage, that means the um, supply of blood and the uh, collection of the deoxygenated blood. The hepatic lymph nodes are present. Okay, mostly round and the portal hepatus. Okay, the celiac nodes and the cytokine chalae. Okay, so the innervations like the hepatic plexus and the cerebral plexus are present, that these plexus nerves having the uh, neural connections. Okay, so the clinical relations, the liver performs the hepatic failure like cirrhosis. Okay, in case of the those persons who are drinking the alcohols. Okay, then the portal hypertension will be there, or the cholestasis will be there. Okay, then hepatocellular carcinoma, the cancerous conditions are also there. Then the hepatitis A, B, or B or C also having the clinical relations with the liver. Okay, so we will stop here and we will meet in the next session for the next gland, that will be nothing but the pancreas. Okay, thank you.